ഹായ് എവ്രിവൺ. ഇന്നത്തെ വീഡിയോയിൽ നമ്മൾ ഡിസ്കസ് ചെയ്യാൻ പോകുന്ന ടോപ്പിക് ആന്തർ കൾച്ചർ ആൻഡ് പോളൻ കൾച്ചറാണ്. അപ്പം all of you definitely know what is an anther and what is a pollen, right? So in this figure we can see a cross section of a flower demonstrating different parts of a flower, and you know that this is the anther and this is its filament, and this anther with its filament is shown here. So anther, this is the anther lobes and it has a filament. So both these are together called as stamen. That is all just basic things, and I'm not supposed to explain all this, but still to clear the concept. So here we are culturing this anther or the pollen grains which are formed inside the anther. So here you can see the pollen grains. Pollen grains are found inside this anther lobes. So we can culture either the anther or we can culture the pollen grains. So these are cultured for developing haploid plants. Okay, so the question will be coming like haploid production through anther culture or haploid production through pollen culture. So you are supposed to answer the question by describing the anther culture process along with the flowchart or diagrams. So anthers are cultured in lab conditions or in vitro conditions in a suitable culture medium for developing haploid plants. So the development of haploids through anther culture is called as androgenesis. Okay, so the regeneration of plants from this anther culture is called androgenesis, and the haploid plants which are produced by anther culture are called androgenic haploids. Okay, and first these type of cultures were, or these type of experiments were carried out by Shimakura, a scientist, in 1934, and later Indian scientist Maheshwari and Guha in 1964. They went for culturing the anthers from the plant Datura inoxia, and they produced embryoids to develop plantlets. So they were the pioneer scientists who were working in this haploid production, Maheshwari and Guha. Later haploids were produced from various plants like potato, pepper, wheat, tobacco, etc. And in anther culture, the overall, you just remember this flow chart, you will be clear with the concept. So we have the flower buds. This flower buds contains anther, and inside this anther there will be microspore mother cells. Microspore mother cells undergo meiosis to produce microspores, and these microspores are formed in tetrads. That is, the pollen grains are formed in tetrads. These tetrads, when they undergo division, it may be either uninucleate or sometimes it may be binucleate. Okay, so both may be the condition. In binucleate, if the pollen grain or the microspore is binucleate, it is inoculated to a culture medium and it undergoes division to produce embryos, and from that embryos plantlets will be developed. Or if it may be uninucleate, that also may be cultured in a culture medium to produce a callus, and callus undergo embryogenesis to produce embryos, and embryos are then regenerated to plantlets. These haploid nucleus or haploid microspores, they may be induced to form binucleate condition, homokaryotic binucleate, by a mutagen known as colchicine. So colchicine treatment makes them binucleate, and these both nucleus fuse to form a diploid cell, and this diploid cell undergoes again division and forms callus, and from that callus embryos are formed. That embryos will be formed, regenerated to a plant. So this is the overall process happening. So what we do, or what is the procedure we do for developing a haploid plant? So we have a plant here. The plant is selected, and from that plants we select unopened buds. Okay, so we select unopened flowers, and these flower buds, they are washed with distilled water, then they are dipped in 5% T-pole solution, or you can do this process with any other detergents, then again washed with distilled water. After that we move to laminar airflow chambers. Inside this laminar airflow chambers we do surface sterilization with the help of sodium hypochlorite, or we can use 0.1% mercury chloride also. So surface sterilization inside this laminar airflow is done with either 5% sodium hypochlorite or mercuric chloride. And after surface sterilization with these chemicals, we wash these flower buds again with double distilled sterile water, and this sterile flower buds are then cut to separate the anthers. So anther or ovary, whatever we use, it is separated. Here we are particularly mentioning about anther or pollen culture. So instead of separation of ovary, you can write separation of anthers. So from this flower buds, anthers are separated, and these anther buds are inoculated to culture mediums. So either in petri dish or in a culture tube, we inoculate these anthers or pollens, and then incubated in alternating light and dark periods, 16-hour alternate light and dark periods in 25 degrees Celsius controlled conditions. And after 2 weeks we can see a plantlet will be it's not like every plant will be regenerated after 2 weeks. Initially they may be producing a callus, and from that callus embryos will be produced if we go for indirect method. Sometimes directly a plant may regenerate from these anthers. So anyway, after a particular time we can see that a haploid plant is regenerated from these anthers or pollens. So this is the overall procedure we are following. And coming to the particularly the pollen culture, in case of pollen culture we have a sterile flower bud which we have selected from these processes. Flower bud is selected, and all these processes are common: washed with distilled water, detergent, and again washed with distilled water, and then they are cut from their filaments. Anther lobes are separated, and they are put in a culture or tube or a test tube, and then we squeeze these anthers gently with the help of a scalpel. So by this squeezing we can either use a glass rod. So with the help of a glass rod we squeeze these anthers inside this tube, and this helps to release the pollen grains inside these anther lobes. So pollen grains will be released, and after that we filter these squeezed material so that we can separate the debris of anthers, and we get only the pollen suspension. So we have the filtered pollen suspension, and these pollen suspensions are centrifuged to get the sediment. So we need only the sediment. This pollen we use for culturing. We culture it either in a solid medium or in a liquid medium, and then in MS medium we inoculate them and incubate for 2 to 3 weeks, and after some time we can see the development of embryoids. Callus will be developed and embryos will be developed from that, and these embryos are transferred to subsequent mediums for induction of shoot and root, and we can regenerate haploid plantlets. Okay, because these plants are regenerated from these pollens. So pollens are the gametes, so gametes are haploids. That is why we are getting the haploid plants. So regeneration of plants from the gamete cells like pollen or from the ovum, that plants will be haploid. So this is how we can regenerate haploid plants. So I hope you are clear with this concept.